Hey everybody, my name is Jason Walker and I am a science coordinator with the National Math and Science Initiative, and this unit is all about biotechnology. So we're going to be dealing with DNA transfer and different processes in which that DNA transfer can occur, causing an increase in variation in a particular population of organisms. Okay, we're going to be talking about viruses, we're going to be talking about bacteria, we're going to be talking about genetic engineering. So even we as humans are facilitating processes that increase variation and cause changes in the genomes of particular strains of bacteria or other organisms. So let's start off this particular presentation that's called Increasing Variation Through DNA Transfer. So let's start off just thinking about antibiotics. Okay? So have you ever taken an antibiotic? Why do you think that it's important that even when our symptoms are gone, we still need to take the rest of the prescription? And also think about, do you think that we are overusing antibiotics? Let's go back to the evolution unit. Let's think about the bottleneck effect, which is just a type of genetic drift. Some of these bacteria are going to be resistant to the antibiotic, right? So let's think about all that antibiotic goes in there, it starts killing off all of those bacteria. Well, some of them are just naturally resistant, or they have some acquired resistance to it. They survive, right? And then they're going to repopulate. Well, every time you bombard that with a new antibiotic, the bottleneck effect occurs over and over and over. So we are facilitating natural selection on these strains of bacteria when we do this, right? So let's think about this as we start talking about antibiotics. First of all, let's just get this out of the way. How does a bacterium actually fight off an antibiotic, right? What is it that they are doing that we say, okay, they're resistant, right? So three main ways. The membrane, either the bacterium has membrane proteins called efflux pumps that literally just pump those antibiotics back out of the cell, right? Or the prokaryote can produce an enzyme. Now, this enzyme can either do one of two things. It can either degrade the antibiotic, destroying it, right? Or it can change the structure of the antibiotic. Now, remember, in biology, this is huge. This is a huge theme here that we have to get. This just flows all the way through every chapter, every concept that we deal with in biology. When you change the structure, you change the function, right? Change the structure of that antibiotic. You change the function. And now it's not able to, let's say, attach to particular receptors or whatever, right? So how do these organisms become so resistant? Well, three different ways. Naturally, we'll call that innate resistance, right? They were born without certain cellular structures, whether those be transport systems or membrane receptors, that these things are needed for the antibiotic to be effective. Or, number two, they acquired the resistance post-birth, right? The organism obtains the needed resistance gene, often a plasmid, okay, from another source, which it incorporates into its genome, and guess what? Now it's resistant, right? Okay, so let's talk about the third thing. Just random mutations. Just random mutations that might allow it to become resistant, right? It has been estimated that 1 in every 10 to the 8th or 10 to the 9th bacteria will develop resistance when exposed to an antibiotic. Now, this might seem extremely rare, but let's think about this for a second. If we start with one bacterium, and if that one bacterium, let's say that it's like E. coli, which under ideal conditions can replicate every, let's say, 17 minutes. But let's just say, due to certain conditions, this thing is going to every 20 minutes. It's going to replicate every 20 minutes, okay? So we're going to start off with one bacterium. It's going to replicate. It's going to undergo binary fission every 20 minutes for 24 hours. So we start off with one. And 24 hours later, we have over 4.7 times 10 to the 21st bacteria. Now that is a bunch of bacteria, right? And if we think that 10 to the 8th to 10 to the 9th bacteria will develop resistance when exposed to an antibiotic, that's a tremendous amount of resistance. A tremendous amount of bacteria that are not affected by the antibiotic that we just put inside our body, right? So when they have this new gene, whether they come by it naturally or they acquire it, or the resistance, that new gene is acquired just due to random mutations. If that is then given to the new generation, to the next generation, with the next generation, with the new progeny, this is called vertical gene transfer. Okay? So let's talk about horizontal gene transfer. That was vertical, one generation passing it, that particular resistance gene, to the next generation. Let's talk about horizontal gene transfer. There's 4 different ways that we're going to talk about here where genes are transferred from, like, let's say, one organism to the next. So it's not a reproductive event, right? They're getting it from the environment, or from another bacterium, or from a virus, or whatever. Okay? So let's talk about this. Transformation. Transformation is the uptake of naked DNA, usually a plasmid, by a prokaryote. Right? You may have performed the transformation lab with your teacher. Okay? You introduce this particular plasmid to a bacterium, and it just incorporates it into itself, right? So what's transduction? Transduction is the viral, so now we're talking about viruses. So viral transmission of genetic information. Conjugation is when one bacterium just says, hey, I'm resistant, and I'm going to copy my resistance plasmid. I'm just going to give you a copy, and that is going to make you resistant as well. That's conjugation. We'll talk about that, look at that here in a little bit. Finally, transposition. Transposition is just the movement of DNA segments within and between DNA molecules. Okay? So let's look at this slide here. This is an extremely famous experiment, okay, that was performed by a guy by the name of Frederick Griffith. Okay, and he performed this experiment to show how heat-killed virulent strains of Streptococcus pneumoniae conferred their pathogenicity to non-virulent strains. So let's look at all 4 different experiments that he did. The very first thing actually the very first 2 we want to look at as controls. Okay, actually the first 3, they are the controls. And the actual experimental group was just this last one. So let's look at this. In the first one, he gave mice living S cells which contained an outer capsule that protected the cells from the immune systems of the mice. Okay, these mice died. Okay, so how is this a control? Well, this is a control because if you give mice living S cells, they will die. Okay, so let's look at what he did to the next group of mice. He gave them living R cells. Now, the R cells did not have the capsule. So when you injected the living R cells into the mice, their immune systems, because the R strain doesn't have a capsule, were able to fight off that infection, and look what happened. They lived. Well, the third experiment that he did, actually the third control, okay, is he took some S cells and he used heat to kill them. He injected those heat-killed S cells into mice. And look what happened. they lived. They were healthy. Now, here it is. In the next group of mice, he injected a mixture of heat-killed S cells and living R cells. Okay? The mice died. Okay? So let's look back. If you inject living R cells into mice, they live. If you inject heat-killed S cells into mice, they live. But if you inject both of those together into mice, the mice died. So what happened? What he concluded was that the non-virulent cells, which were the R cells, received the pathogenic substance from the killed strain. So even though Griffith killed those S cells, the R cells were able to get the DNA that allowed the R cells to make that capsule. Because they were now able to make that capsule, because of that genetic information they got from those heat-killed S cells, they were able to make that capsule. The mice, their immune systems were not able to fight off those R cells because it had that capsule, and they died. Okay? So now, let's talk about this. You have R cells that now have new genetic information in them, right? This causes the cells to become what we would call a recombinant cell because it contains genetic material from more than one source. Now that's transformation. Okay? Transduction is the process that a virus is carrying prokaryotic genes from one bacterium to the other. Okay? So let's look at what's going on in this picture and just remind ourselves of what we've studied back when we studied DNA. This is the Hershey-Chase experiment, right? When they were trying to figure out what is the genetic material. Is it protein or is it the DNA? Okay? And so they actually used viruses. They radio-labeled the viruses, okay? And they radio-labeled DNA to see which ones were actually being transferred from the virus to the prokaryote, inside the prokaryote, right? And what they figured out was it was actually the DNA, not the protein, okay? So, but it was transduction, the process of transduction, where viruses are able to take bacteria from one bacterium to the next. This whole process, because we knew that that's what viruses were able to do, it allowed Hershey and Chase to perform this experiment. Okay? Okay. So let's look at transduction just a little bit more in depth. Okay? So first you have this bacteriophage. You have this virus that is going to attack the bacterium. So this bacteriophage injects its genetic material into a particular bacterium, into the cell. Right? Let's look at what happens. The bacterial DNA breaks apart. The viral DNA uses the cell's machinery to make more viruses. Right? Now remember, what is a virus? A virus is basically just genetic material with a protein coat around it. Essentially. Right? And so, when that virus injects its DNA, the cell has the ability, it has the enzymes and everything that it needs. It has the machinery to take DNA, use that to make RNA, then use that RNA to make what? Protein. Transcription and translation, right? So essentially, all you need to do is to make that outer capsule, make the protein, incorporate the DNA within that, and you have the virus, right? Well, sometimes Some of the viruses end up with the host cell's genetic material, not the viral DNA or RNA. Okay? The virus particles with the host cell's genetic material can then go and inject that into another cell. Now that new cell is a what? A recombinant cell. It's a recombinant organism because it now has DNA from multiple sources. Okay? And this new cell can incorporate this new DNA into its own. So let's look at this and just ask ourselves, how does this increase the genetic variation in a population of prokaryotes? Right? Some of these bacteria are going to have different DNA now than their brothers and sisters, cousins, uncles, grandmas, and grandpas around them. Right? Well, let's look at a way that maybe some of those bacteria with the new DNA within them, some of these recombinants, can then confer that to another bacteria that doesn't have it. Let's look at conjugation. Okay? One of the bacteria, okay, it sends out what we call a sex pilus, which attaches to the other one. Okay? It's then going to pull those two organisms together. A mating bridge is then going to form, okay? And it allows new information, that genetic information we just talked about, to be passed from one to the other. Okay? So here we have, this slide shows two different ways in which bacteria can use conjugation to transfer a gene to another bacteria. So in A, we see A shows a bacterial cell that's transferring an F plasmid. Okay? Now this plasmid confers the genetic information that's needed to make a sex pilus. Once this process is complete, the cell that just received the new F factor, or this gene, can produce a sex pilus and give the gene to another cell which is negative for the gene. Okay? Now one thing I would like to stop and just talk about real fast is, if we look at this F plasmid right here, you see how it's all dark blue? Okay? It's going to make a copy. It's going to make a copy where and send that copy to the new cell. Right? But I want you to look real close at this quote-unquote copy that it's sending. You see, this is all dark blue, but as it makes a copy, the copy is light blue. And so what you end up with is a semi-conservative model. Do you remember when you were studying about how DNA replicated, and we said that you have two strands of the DNA, right? That unwinds. DNA replication occurs, right? So DNA polymerase is going to come and bring free DNA nucleotides, right? The one with the guanine, one with the cytosine, one with the thymine, one with the adenine, right? And it's going to make a copy of each side, each strand. So when you end up with two new strands, two double-sided strands, each will consist of one strand that is the parent strand, and the other one is the new copy. Right? The same thing is happening right here in the bacterium. Okay? We call that the semi-conservative model. So if we look at B, well, how is B? Look at B. B is different than A. How is that different? Okay? That shows, B is going to show part of a cell's genome from a recombinant cell that contains genetic information from two different sources. So this is already a recombinant. Okay? And this is being copied, and it's transferred to another cell which is not a recombinant. This new cell is now a recombinant. Okay? So let's take a look at this slide. Let's just think to ourselves. Okay? Can we explain how this process in this slide increases genetic variation in the population of prokaryotes? Think about that for a little bit. Just in one slide, what I wanted to do is just put what we've talked about so far. Transformation, okay? It just involves the uptake of short fragments of naked DNA by naturally transformable bacteria. So it's not conferred from another bacterium. It's not necessarily. It's not brought by viruses. It is just there. We as humans can do this, right? In the transformation lab in your AP biology class, you do this. We can force transformation to occur, right? So let's look at transposition. Okay? Now, these transposons have been characterized as what we call jumping genes. Okay? Basically, you just have small DNA segments that can be transferred from one place on a chromosome to another. Okay? And basically, this is done by what you might recognize as a cut and paste or a copy and paste. Okay? We also have retrotransposon movement. This is when an RNA intermediate is made and is used to make a new DNA segment using reverse transcriptase. That DNA is then inserted elsewhere in the genome. Okay? Now in 1983, at the age of 81, Barbara McClintock won the Nobel Prize for her discovery of these jumping genes in corn. Okay? Now you can see how these transposons have varied not only the genotype, but also the phenotype, right, of this corn cob. So when you are looking at, let's say that you have a corn cob that is the progeny of a dihybrid cross between two heterozygotes. Let's say, and some of you may have performed this particular lab in your pre-AP or your regular biology course, or maybe even your AP biology course. But basically, you have a corn cob that has four phenotypes: smooth versus wrinkled is one trait, and the other trait is either it's yellow or dark. Okay? So there would be four different phenotypes if you had two heterozygotes. Okay? You'd have smooth wrinkled, you would have smooth yellow, smooth dark, wrinkled yellow, and wrinkled dark. Okay? Now, you know in a dihybrid cross between two heterozygotes, you should get a 9:3:3:1 ratio, right? You're going to have, out of 16 squares, you're going to have 9 that are dominant-dominant, 3 that are dominant-recessive, 3 that are recessive-dominant, and then 1 out of 16 theoretically should show both recessive phenotypes, recessive traits. But think about what transposons would do. I mean, look at these corn kernels. Are they light or are they dark? Well, they're actually neither, aren't they? How would you count those? So you can see how we started off thinking, okay, I only have 4 different phenotypes that I can look at for each individual corn kernel. But that's not right. Right? Transposons have caused some of these to change genotype, change phenotype.